Mutations are a fact of life, and in fact, if it wasn't for mutations, there wouldn't be any evolution. But for this part, what we're going to concern ourselves with is what are different types of mutations and what are their consequences on a protein sequence. So when we talk about mutations, we're referring to a change in the amino acid sequence. So let's find out and learn about different types of mutations. We're going to start with this sequence on the top. That's our starting sequence. And we're going to look at, first, we're going to look at point mutation. That's where a nucleotide, a single nucleotide, is replaced with another one. So when we substitute a nucleotide with another one, there's two different ways that it can be done. And let's take a look at this example. So this orange part is the place in the sequence that is changed, and we find the corresponding sequence in here, so corresponding nucleotides. We'll see that a G is replaced with an A. So before we go into the details, let me remind you that a G and an A are purines, and a C and a T are pyrimidines. So if you replace a purine with a purine or a pyrimidine with a pyrimidine, we call this a transition. So a change of a purine for a purine, or substituting a pyrimidine for a pyrimidine, that's called a transition. Then we have transversion, which is when you change a purine and replace it with a pyrimidine. So again, in here, if we look at the G in here, G is purine, and when we go look in here, it's replaced with a C. So we're changing a purine with a pyrimidine. Or in here, if we look at the bottom strand, we are changing a pyrimidine C to a purine A. So that's a transversion. Then we have more gross changes in the DNA sequence that are not point mutations and so they're not substitutions. You can have big chunks of a DNA deleted, or sometimes even small chunks, or there are sometimes insertions when you insert a sequence into a DNA sequence. So what is the consequence of these events? So for point mutations, there could be different outcomes. So if we replace, so let's see which one we're replacing here: serine, valine, proline, and tyrosine. And the one that we are changing is this last position on valine. We're changing it from an A. I think I just covered the A. We're changing from A to T. So this is a transversion. We're changing a purine to a pyrimidine. And you can see that there is no consequence to the amino acid sequence. This is still valine. It was valine and it's still valine. That's the amino acid that corresponds to this. So why does that happen? Well, remember we said the genetic code is redundant. So there are multiple ways to encode a particular amino acid. This type of a point mutation is called the silent mutation because it really doesn't make any difference in the amino acid sequence. And you can imagine that the protein product also won't have any effects on its function. Then we have a mutation where if you change it, you actually change the amino acid. So in here, we're starting with proline and we're changing it to threonine. Proline is a hydrophobic amino acid. Threonine is polar. So how does that happen? This first letter of the triplet is changed from a C to an A. So that obviously changes the amino acid sequence, and totally you get a different amino acid. And that's called a missense mutation, which results in an amino acid substitution. Sometimes a change in a nucleotide doesn't give you an amino acid, it gives you a stop codon. So there's not going to be any amino acid at this position because of the change of this C to a G. So basically, there's nothing in here. That's why this type of mutation is called a nonsense mutation. You substitute a stop codon for an amino acid. Now, another form, so when these are all point mutations, when you have changes, gross changes in the DNA sequence, you can have really grave consequences on the sequence of the amino acid and totally change it. This type of mutations are called frame shift mutations because it totally changes the reading frame of the protein. So in the case of a frame shift mutation, you can see that in this current reading frame, which is the normal sequence, we're going to read these triplet codons, A, G, C, CTA, CCC, and TAC. But in here, after the valine codon, after this G, all of a sudden, we're going to shift the frame shift as if these two have been deleted. So if we delete these two sequences, then you shift the sequence down. Now it doesn't make any difference for valine, but look at the amino acids because of that redundancy situation. You can see that you're totally changing the sequence of the protein. You're not changing just one amino acid, you're changing a whole bunch of them. Now this frame shift mutation can be really deleterious. So what is a reading frame? So if we have, let's say, ATG, and just I'm making up something in here, CCC, and let's just as another example for you, let's say TTA. Okay, now let's say I delete this C from here. Originally we would have ATG, CCC, and TTA. But what happens if I delete this C? Then I would have my reading frame would be ATG, CCT, and then TA, and what's the rest of it? So let's say there was another A in here, then it's going to be TAA. The sequence is completely different. Another way I can do this is by adding a nucleotide. Let's say I add another C here. So let's say I'm adding let's say a G in here. So if I add a G in here, what will be my reading frame? It's still going to be ATG, GCC, CTT, and then AA something else down the line. So removal of nucleotides and addition of nucleotides really messes up with the reading frame of the protein and really destroys the sequence of the protein and ultimately its function. So I have at the end of the slide some problems that you can use to test your understanding of the material. And here I've covered the steps of the translation initiation. See if you can draw these from your memory. In here, these are steps of translation elongation. See if you can fill these boxes. This is the Hershey and Chase experiment. See if you can answer the questions shown in here and explain why. And then this is the Frederick Griffith experiment, and see if you can reproduce the experiment and explain what's happening in here.